Hello, my name is Dorothée Sturm from the Paul-Langerhans Institute at the University of Technology in Dresden. Laser microdissection is a technique that allows to obtain a special cell population from very little amounts of parenchyma. The dissected cells can be used for transcriptomic and proteomic studies, DNA assessment or chromosome analysis. Here we provide our protocol for acquiring human pancreatic beta cells from surgical specimens to be used for transcriptomic studies. Cut the pancreatic tissue into small pieces. Place one piece in the center of a cryomold and cover it with chilled embedding compound. Freeze the tissue immediately with chilled 2-methylbutan and store it at -80 degrees Celsius until sectioning. Label 40 adhesive slides and pre-cool them inside the cryostat chamber. Fix the frozen